talk ions and mechanisms in the coronavirus replicase. So, shown on the screen, we've got the entire coronavirus replicase, and I've got kind of a rundown of the ions. This is like a list, and what I'm actually going to be looking for is, can you go beyond this list and tell me like why and what are these things doing? That's like full understanding of the enzymes at an enzyme level. So, to overview, there are lots of ions that are associated with different parts of the SARS coronavirus replicase. And so we're only counting the conserved parts that are there in every single coronavirus we've found so far. And we'll put N-endo U there, even though it's missing from a couple coronaviruses, but it's pretty well conserved. We've got them all on here. NSP3Y domain has not yet been solved, but we know there are two zinc finger motifs in there. And from personal experience, I know that if you try to alter those, like mutate those, you get a dead virus. So they're important in some way, but we can't say exactly how. There's a structure that doesn't show the metal ions that shows roughly where they are, and they're down right by the plug of the DMV pore. Okay, NSP 4, 6, and 7 definitely, as far as we know at the moment, do not have any metal ions associated. NSP 5, there is one paper that shows that in one form of the purified protein, there's a zinc which is actually bound to, interacting with one of the catalytically important cysteines in the molecule. It is not certain and is actually not thought probable that that's actually part of the enzyme mechanism, but it's there, so I'm telling you. So, maybe, but probably not. Let's see. NSP8 now, that is an enzyme, and that is going to be... It's part of the machinery that makes the polymerase work, but on its own, NSP8 has a little bit of polymerase activity. Seems to really like making poly A's from a poly U template. So it may have a role in polyadenylation of the plus strand 3' ends, but that is not certain. This is like an isolated enzyme with part of the complex. We don't have full working details of the whole thing. Anyway, that thing requires one divalent cation, and what you'll see throughout here, wherever it says magnesium, it'll usually work pretty well with a manganese. In this case, we do know that if you put in manganese, it makes more mistakes. And that's pretty common in polymerases in general. It just gets a more specific hold on nucleotides, and they have to match the opposite nucleotide a little bit better if there's magnesium there, as a kind of a general rule of thumb. Yeah, I'm sure there are exceptions, but it's a good start. All right, NSP10 is going to be a cofactor for NSP14 and NSP16. So there's separate NSP10s interacting with both. It's not one doing double duty. It's needed for at least some of the enzymatic activity of NSP14 and the only enzymatic activity of NSP16. That's got two zinc binding sites, and shown on here, these are all zinc binding sites, but everywhere you see this little orange and yellow blob, that is a zinc binding site that can at least sometimes be switched out for an iron-sulfur cluster. So like NSP10, they're probably zincs, they look like zinc fingers, but those same cysteine and histidine residues would coordinate an iron-sulfur cluster, and that may actually be the preferred substrate. The jury's a little bit out on that right now. We'll come back in a couple of years and hopefully know which one is more important. And it may be that it works with both. I have no idea. All right, NERAN, which is going to add the first nucleotide to NSP9. So NSP9 doesn't require an ion, but NERAN does require an ion to do this. And it's actually got two catalytic divalent cations with a preference, we think, for magnesium. And those are going to do the standard thing. They are going to sort of pinion the oxyanion on the phosphate of the nucleotide that is going to be attached. And this nucleotide is going to become the first nucleotide in the genome, and then later it's going to come back and it's going to add the cap nucleotide on. It's the same active site that's able to do both of these things. It's just got different binding pockets for the NSP9 and the first nucleotide and the cap nucleotide. So as far as we know at the moment, it's adding them the same way, but there is definitely more to that story that has to be learned. The RNA polymerase has a couple of zinc binding sites that can also be substituted out for iron sulfur clusters. So that's the two that are shown with the blue domain, which NSP12 is the RDRP. And it also has two catalytic magnesiums that are going to pin the oxyanion just like they always do in polymerases. So this is going to stabilize the incoming nucleotide to allow a nucleophilic attack to actually join the nucleotides together. That's the special environment that the polymerase provides that makes catalysis possible. Okay, helicase. We know it requires magnesium. There's at least one structure that shows a magnesium in the active site. So this domain is going to, or this protein rather, is going to have two domains that are called Rec-A domains. And if you imagine those are a butt, the crack right between those, that is where the active site is going to be. And that is where the magnesium is going to be coordinated. So it's involved in the active site, and there's also a phosphate ion that sits somewhere around there in the crystal structures. We don't know if that is a thing that has to be there in the working enzyme, or if this is just a thing that you get when you crystallize it. It's not clear to me anyway. Let's see. Next up, helicase is going to have... Oh, no, we already did helicase. Sorry. Next up, the exonuclease and N7-methyltransferase. So those are the two halves of NSP14. There's 3 metal ion binding sites on those. 2 of those can be substituted for iron sulfur clusters, 1 on each domain. There's also a zinc binding site that's kind of at the junction of the 2 domains, because it's like two blobs that are next to each other with a little chain in between them. That's what the protein looks like. So each domain is like an enzyme all by itself, and if you cut them apart, they could almost be an enzyme, except that part of this enzyme is going to actually push on this enzyme to knock it into the right shape so it works. So they don't really work on their own as far as I'm aware, but they have all the components that they would need to work once they're in the right environment, which is being next to the other half. Okay, so that's going to have an active site magnesium or divalent cation, and it's going to have a couple of zincs, one of which is at the junction. So I put it with XON, but you could say, well, it's halfway attached to N7-methyltransferase, and you would not be wrong. N7-methyltransferase either has a catalytic zinc, which would be weird, but may just be there to position the nucleotide, like for methylation, or it may have a catalytic magnesium. I have not seen a convincing study that is certain of this. Like sometimes when you crystallize these proteins, they come down with ions that are a little weird, and it's hard to tell if that's the needed ion in actual catalysis. Let's see, the endonuclease. So the exonuclease is our proofreader. The endonuclease is going to trim off the 5' end of the minus strand, which is a bunch of U's, and it's going to trim it off in such a way that it doesn't leave a free 5' phosphate. And so that has a catalytic magnesium that's going to do the job. And then the 2' O-methyltransferase uses a magnesium to orient the nucleotides so that it can add the zinc to those. The folds are pretty similar. They're very similar enzymes, the N7 and 2' O-methyltransferase. They're just methylating at a different position on the RNA, and it's a kind of a similar mechanism, which is why we think they probably work the same and have the same cofactors. That would make the most sense. Not guaranteed, though. Whoa. And let's go back to me. Sorry about that. Yeah. Okay. Let's look at them one at a time. All right. Here's the first one. This is the NERAN domain. Here are the two magnesiums. It's these nice little yellow dots right here. This also shows that if you feed UMP and NSP9 there, it will put all the UMP onto NSP9. It really loves doing that. But it's also pretty good at adding any of the nucleotides if you only give it one nucleotide as a choice. So this is like the order of preference. U is greater than A is greater than C is greater than G. But if all it's got is a G, it can add a G if that is necessary. And there may be some template dependence for this, because depending what nucleotide it adds, it's going to pair up with one end or the other of the template, depending whether it's the positive or negative strand. All right. Here is the RDRP, and there are the two they're showing them as manganeses because that's the way that the crystal structures come out best. But they seem to also tolerate magnesium, and that's probably the preferred ion. It's a little more common in nature, and is more common in these structures. And there you can see they've actually superimposed the active site of the coronavirus, RDRP, with the active site of the hepatitis C virus, which is a nice flavivirus. And you can see that pretty much everything lines up in the same positions, same side chains, same two divalent cations. Really a very similar structure. And they're also showing the position of several inhibitors, how they bind to it. And they're all interacting right at that active site. And you can see the phosphates here are what's, that's this orange and red thing. Those are what is supposed to be interacting with the manganese or magnesium. Next up, we're just going from start of 1B to the end, looking at the enzymes. This is the helicase, so it's got this zinc binding domain. It's actually got 3 slots for divalent cations, or one of those can be a iron sulfur core. And then it's got what's called the NEC domain, and then it's got this little 1B domain right there. And then right at the end of that, that's where it's going to have the Walker A motif, which is going to bind ATP. That's going to power the process, or at least the helicase activity. Over here is the active site, and that's where it's going to have the metal ion. That's our little green dot. And this one was solved with magnesium. That seems likely, but may well tolerate other ones. Okay, NSP14 with its cofactor NSP10. So 10 is the pinkish red thing that's over on this side. The yellow middle bit, that is going to be the exonuclease, so the proofreading enzyme. And then the blue bit that's over here, that is going to be the N7 methyltransferase, and they're showing the methyl donor, I believe, in there. I think that's a SAM molecule, because it is a S-adenosyl methionine dependent enzyme. So you got 2 zincs in NSP10 that can be substituted maybe for iron sulfurs, but I'm going to stop saying that because I've said it enough times. You either got it or don't. Here's the active site magnesium of the exonuclease, and so this is going to be part of the exonuclease mechanism, and it's going to be driving an attack, basically, that is going to end up breaking the phosphate linkage, and that's how you're going to remove the, it's the last nucleotide, the 3' most nucleotide that was added. Here's the zinc binding site of the exonuclease. Here's the zinc at the interface. You see it's interacting with the methyltransferase and with the exonuclease. And here's the zinc binding site over here that's just for NSP14, helps it fold up as far as we're aware. There you go. So that's a capping enzyme. All right. Next enzyme, not universally conserved, but so close that it might as well be. this is the endonuclease NSP15. So this can cut in the middle of an RNA chain, so potentially dangerous, but it has a real preference for lots of U's, or like GUU. I think it was OK cutting in the original paper. Cuts by a weird mechanism though. So it's got a divalent cation. That's this B with the 2 electrons here. That's what I'm circling. That is going to stabilize the hydrogen ion for a sec. That's going to give oxygen back its electron. It's going to then use that oxyanion electron to basically stab the phosphorus, the phosphate. And so we're using, this is the 3' oxygen, the 3' hydroxyl before it was added. This is the 2' one. So this is the one that's on RNA and is not on DNA. And so we're going to use the 2' to attack at the phosphate. And when it does that, it's going to make this weird little ring. And so the phosphate is going to be left here attached to both the 2' and 3' hydroxyls. That's weird. And this is the phosphate that should be on the 5' end of the other nucleotide. But you can see the 5' end of the other nucleotide just gets an O-, which is then going to pick up a hydrogen to become an OH. So you end up with a 5' hydroxyl on the cleaved product, which is not a thing that is easily recognized, I believe, by RIG-I and MDA5. And so that essentially becomes invisible. So this is a modification to the 5' end of the coronavirus minus strand, which is poly U, which is going to shorten it a little bit, and it's going to leave it with 5 to 6 U's, and those the last U in the chain is not going to have a phosphate on the end. No free phosphate. It is going to have a hydroxyl, and so that helps to evade recognition by the intracellular immune pathways that are looking for miscapped or uncapped RNAs, which are a thing that viruses do sometimes because they're sloppy, and not a thing that the cell would normally do at all, really. Last enzyme. Sorry, I'm balancing on a coffee cup. Yeah. So this is the methyltransferase, and again, it's got an NSP10 cofactor. That's the pink thing at the bottom. The methyltransferase itself is the big blue thing at the top. That red thing in there leading up to the gray, that is the chain. The gray is actually the cap, so let's circle that so you can see it, or silver if you like. Silver sounds cooler. The rest of this is the other nucleotides that are behind the caps. We have to have some nucleotides made before this is going to fit into the active site. And it's unclear at what point this is going to occur. Could be the last thing that happens, could happen in the middle, could happen after you add, like, what is that, 3 nucleotides plus the cap? Yeah, hard to say. This is going to have an active site. The active site is going to have a magnesium ion, Mg2+, and that's going to be necessary for catalysis. And then there's NSP10 with its double zinc finger. Yeah. And so it's holding on to stuff and doing stuff. Yeah. Being the best NSP10 that it can. All right. I think that's all of our, that's all of our little enzyme mechanisms. Just some extra detail that you can hang on your answer and make it really molecularly correct and good. Yeah. All right. Hope that helps. Thanks.